This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 887, recorded on April 8th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello there, Vincent, and all the ships at sea and everybody else. Looking out my window and having it open for most of the day let in a most pleasant weather pattern. It's a Georgia O'Keeffe afternoon. The puffy white clouds are all about even sized. It's about 68 degrees, light wind, beautiful, beautiful, beautiful day. Wish this podcast were being held outdoors, but it's not.
Dixon, today is nice and sunny, as you know, as you just said. And we had two previous days of cloudiness, and it just emphasizes how much better your psyche is when it's sunny.
No question. In fact, we had flooding rain last night in New Jersey.
Oh, we did have flooding rain. Also
Crazy.
Joining us from Austin, Texas, Rich Condit.
Hi, everybody. 76 degrees and sunny, also gorgeous. It's a beautiful spring day here in Austin, Texas.
There you go. The bluebonnets are out?
Oh, yeah, including in our front yard.
Well, all right then.
And from New York, Amy Rosenfeld.
Hello.
Hey, Amy.
Oh, it's a look like out the window there.
You got some news for us?
I got white puffy clouds, and
You got the ones I see.
I was going to say, and it's my favorite time of year because it's the Masters.
What else is new, Amy? Come on, tell us.
What else is new?
Tell us.
I think Tiger is screwing it up.
No, that's not what
He
I
is.
was.
+2.
I thought you said two. What does that mean? 2 over par?
2 over par. Aye.
Okay. Amy, what's your big news? Or is it too early to reveal?
He doesn't want to reveal it. It's okay.
I guess not.
However, I do have some other news. Tomorrow is the birthday of Malika, who is a listener and is currently in a PhD program and very interested in working one day on vaccine development. She believes strongly in global vaccine availability and equity. Her favorite group of viruses are those that cause hemorrhagic fevers. So we'd like to wish her a happy birthday and a successful PhD. And we're sure that, I'm told that her passion is incredible for viruses. And so that'll be a good quality to bring you through.
Where is she in school?
PhD school. Oh, where? I don't know.
Ah,
I wasn't provided that information, but wherever you are,
right.
all the ships at sea, as Dixon said.
That's right.
Happy birthday, and keep on trucking and keep that passion and curiosity. All right,
Keep on placking.
Oh, that's great, Dixon. Keep on placking. So that's like Keep on Trucking, which was a Grateful Dead song, right?
That's right. I even have a picture of 3 elephants, the mama, the papa, and the baby.
Yeah.
And it's entitled Keep on Trunkin'.
Huh. Someday we have to use that in the title, don't you think, Rich? Keep on placking.
That's right.
Placking with an N and a little apostrophe to show that the G is gone. Right?
That's right.
Because that's what trucking was. Oh, that's good. Dixon, it's always a pleasure. You're so creative with this cultural stuff, you know?
Someone has to make up for the missing Alan Dove, who would have gone way beyond that, I'm sure.
It's a good team.
Seems to be missing in action many days.
Indeed.
Yeah, I don't know what's happening, but I do miss the members of the crew. But, you know, that's the cool thing about TWiV. We have a large cast.
We do.
What do they call the when there's a leading person who can't make it? What do they call the person who's
the stand-ins?
There's a name for that.
Yeah,
Understudy.
The understudy. That's right.
Yeah, we have people who can step in.
Today, the role of Alan Dove is being played by
Dixon Despommier. Yeah, that's
right.
what they do. They put a little piece of paper in the program. He
I
said,
demand my money back.
today the role of Alan Dove is being played by Dixon de Pommier.
That's right, and poorly.
Who is a star in his own right. Yes. I guess the understudies
so then
really...
If he's playing if he's playing Alan, who's playing Dixon?
Oh, that's a good question. He's playing both. Dixon is an interesting character, I have to admit.
My God.
He has a character, which is, everyone has a character, but Dixon has a character.
Really? I'll think about that. I hope that's a good thing.
It's a good. No, you have interesting qualities. It was very clear when I first met you, Dixon. I asked you to do TWiV with me. Okay,
you did, and I unknowingly said yes.
Annoyingly for whom? You or me?
Me. I had no idea of what I was getting into.
You didn't. No.
Neither
but
did
now
Vincent.
that I know. I'm so glad that I've joined this group.
Well, I'm hope you don't regret it because
no. I feel like this is my scientific family.
We take a lot of your time, but one thing you should understand: we enjoy each other's company, but also people who listen enjoy it as well. So
I gather very much so.
It's a pleasing to give people enjoyment as well as educate them. So that's the story. And today we're going to educate you. We have two stories, and the first is all about influenza virus. And I have to say, the impetus for this came Wednesday night. Amy and I do a live stream, and after you left, Amy, someone said, "How's the flu vaccine matching up?" I said, "It's fine," and I don't know why I said that because It's not fine. There's a mismatch, and today we're going to talk about that as a way of making up for that. So we have first a piece in the Morbidity and Mortality Weekly Report, which is called Interim Estimates of 2021-22 Seasonal Influenza Vaccine of United States, February 2022. And they do this every season. They come out with estimates, which they have planned very carefully as to how they're done and so forth. But just to remind you, annual vaccination against seasonal influenza is recommended in the U.S. for everybody 6 months of age or older, unless you have a medical condition that doesn't permit it. And the influenza vaccines we currently use, and there are a bunch of them, they're quadrivalent. They contain typically 4 different influenza viruses. The current one, an H1N1 isolate, an H3N2, and 2 influenza B lineages. And interestingly, so all of those viruses circulate. And depending on the year, one can predominate. This year it's predominantly H3N2. Some years it's H1N1. And why that should be, nobody really understands.
I mean, Vincent, you should clarify circulate doesn't mean that after you inject it, it circulates. It circulates in the waterfowl. Is that correct?
Oh, in people. These are talking about
in people. Just in people. Okay, just in people.
But using the word circulation would imply bloodstream? I don't know.
Usually it does.
Okay. Anyway, so every year, as I said, the CDC wants to know how the vaccine's doing. It's kind of like Ed Koch saying, "How am I doing?" Right? But Dixon, you probably don't remember about that, but
sure I do. Come on.
Yes. So
Ed Koch was the only mayor of New York City who could walk out of a taxi cab without having to bend over.
What are you talking about?
That was another mayor. That was another mayor. I'm sorry.
No, that was Abe Beam.
That was Abe Beam. I take all that back.
It was very short. No,
you can edit that out.
Quite high. It was quite high. Anyway, what they do is they estimate the efficacy or the effectiveness, not efficacy, the effectiveness of the vaccine in preventing laboratory confirmed mild to moderate outpatient influenza. So this study does not address severe influenza or death, although we'll get to that. And what they do is they enroll people in these studies. This is a multi-state study, and then they follow them up. And so I'm going to give you the punchline right here. You
would be a bad joke teller then.
Yeah, I'm not a comedian. I'm a scientist.
It's not a joke.
And this year, through February, October to February 2022, the overall vaccine effectiveness against outpatient, mild influenza caused by H3N2 was 16%. 16%.
That's pretty low. That's very low.
Which is essentially nothing, right?
Essentially nothing.
I'd call that a spoiler alert, not a punchline.
So they say this indicates that vaccination did not reduce the risk for, illness with these predominant H3N2. And they say, though, still you should be vaccinated. And a lot of people are going to ask, so why should I be vaccinated?
That's true.
Because they say that it does prevent serious outcomes. Those are assessed in separate studies, and I'm not aware of any this year, but in previous years, when you have this lack of effectiveness against mild disease, usually it protects against severe disease. So
it's early in the year that probably affects this whole thing, in particular with assessing serious disease. And also, they point out that in the recent past, they haven't even been able to do this because there's been so little flu, they can't even get good enough data.
Right. That's right. COVID's fault, right?
That's right.
Yeah, that's COVID's fault. So this number came from 3,636 children and adults from 7 study sites: California, Pennsylvania, Tennessee, Texas, and Washington, Michigan. And they report when they either feel badly or they have a positive test, and they take the data and they decide whether they find out whether they're immunized or not. None of them get antivirals and so forth, right? So 16% overall. But the predominant H3N2 this year belongs to a clade called 3C.2A1b subclade 2A.2. Now, listen carefully. There will be a test later.
Sort of rolls off your tongue.
2A.2. And this accounts for over 99% of all the H3N2 isolates that have been characterized. They're submitted, many thousands of isolate are submitted every year. And you can actually see the breakdown in isolates at the CDC website. They have lovely graphs about that, but most of them are that 2A.2. And they say post-infection ferret antisera raised against the H3N2 component of the vaccine, which, by the way, the H3N2 component of the vaccine is, they may change it from year to year. It's they don't say what it is here, but I think it's a Cambodian isolate, if I remember. But anyway, if they take that vaccine component that goes into the human vaccine, inoculate ferrets, get antisera, and then try and neutralize these circulating viruses, they take an isolate from a patient of this subclade 2A2, it poorly neutralizes it. And so that explains from a high view why the vaccine's not working, because the antibodies induced by the vaccine virus, it's not subclade 2A.2. Apparently, when they made the vaccine choice, which would have been in January 2021, for inclusion in the vaccine that's produced later that year, it wasn't the predominant subclade. So they put something else in. In the meanwhile, the virus changed, and so now we have a mismatch. And this is, flu is a slippery beast. It changes a lot, and it's as they say, it's a challenge to get the match between the vaccine and the circulating virus.
A shapeshifter.
You think so?
Yeah. I mean, every time you look, it's different.
So now we're already past the time when the recommendations are made for this fall's flu vaccine.
Right.
And in February, WHO has said you need to include the 2A.2 H3N2. So they will. However, what if it changes again, right? Then you're chasing your tail, correct? Is that a good analogy?
Good enough.
Yep.
Yeah, you're chasing your tail. So those are the data, okay? So the miss is not a good, most of the cases are H3N2 and the vaccine is not doing well for that reason. Now, I found while I was looking for information, a preprint from the group of Scott Hensley at the University of Pennsylvania. And Scott Hensley was previously on TWiV. Actually, he's been on twice. And the most recent on 480, where he talked about how sometimes when you grow vaccine influenza viruses in eggs, you select for changes that then are not good for inducing the right immunity in humans. And I found this preprint, antigenic and virological properties of an H3N2 variant that will likely dominate the 2021-22 Northern Hemisphere influenza season. And so this preprint was posted December 2021. And so this H3N2 subclade 2A.2 had already been found to cause, I think it was an outbreak on a college campus last fall. And these students had been vaccinated, and this caused an outbreak of mild disease. So there's already a hint. And so that's why Hanley's paper says this could be the predominant clade. And in fact, it is, right? And so he in this paper, they do some interesting experiments to try and understand what's different about it.
So this paper sort of anticipates what we've just discussed in the MMWR thing. Right?
That's right. So 2A2, go ahead, Dixon.
Yeah, why don't they switch to growing this in plants?
Well, they have done trials on plant-propagated vaccines, so they could, but there's some companies.
There might be fewer changers.
Perhaps, yeah. Or cells. It's harder to grow vaccines in cells than in eggs or plants, actually.
Exactly, and you get a faster result. You could correct it. They had one that I visited down in Texas A&M that was growing the vaccine.
Yeah.
The DARPA had actually commissioned this production, and it was working out very fine as far as they were concerned.
So as long as we've opened up this topic of how you make the vaccines, so making the vaccines on eggs can select for variations that we'll discuss that are not necessarily good, but they're can be ungood, put it that way. So, but there are cell-based vaccines available, correct?
Yes, there are. Yes.
And I also know that there are recombinant vaccines available. Flu block is among these, where the genes are cloned into baculovirus vectors and expressed in insect cells, and the hemagglutinin purified from that. So those are recombinant vaccines. My question is, because I know that there are, at least with a lot of the vaccines that start out with whole virus, these things are passaged on eggs at some point or another. And I'm wondering if the cell-based vaccines and the recombinant vaccines avoid eggs entirely. Seems like they should. Is that correct?
They do. So they should have the right, if their change is selected for growth in eggs, those vaccines would not have them.
So but then the other question is...
There are a minority of the vaccines that are used in the U.S. That's the problem, right?
The other question is that in particular with the vaccine that's a recombinant vaccine made in insect cells, are the glycosylations the same as what you would get, say, from in passage in humans?
That's a good question.
I don't know the answer to that.
Neither do I. We'd need to get Scott. I bet he knows the answer.
Okay. Right. Because the last thing that interests me is that, when you go to your pharmacy to get a vaccine, you're not offered a choice. I mean, there's at least a half a dozen out there.
They don't even know what they're giving you in many cases, Rich.
I always ask.
I do too. They said, I don't know, I just got this one.
Yeah, that's a good point. But you don't have a choice. And I remember, before this year before last, or maybe the year before last, for a couple of years running, I was getting flu block. That was what was recommended for old guys like me, according to the pharmacist. And that's what I got. That's the one that's the recombinant made in insect cells from a baculovirus vector. And then This year, I got something else, a flu quad or quad flu or something like that. The quadrivalent one is recommended for
With the higher
the
dose.
older people, but it's an egg-based vaccine as far as I know. So
Yeah.
I don't seem to have it seems kind of willy-nilly.
Amy, did we get a flu vaccine together this year? I don't remember.
Yes, we did.
So I
But
remember
you did not get high dose flu
No, they
for
said-
old people. They did not have it.
they didn't have it. They
Really?
said, I'm sorry, we don't have the old man vaccine.
Oh, that's nice.
And I
So-
said, actually, they didn't, but I
So
said,
well,
it's
let me
fine.
have
did.
They said, let me have two of the younger ones, then one on each arm. They'll be fine.
I said, give me what you have.
Yeah, and then the following day we came, or 2 weeks later, we came back for our COVID boosters.
Yeah,
And they
so the
still
thing
didn't
is,
have the old people.
The likelihood
vaccine.
that I would go somewhere else to get a flu vaccine is so low that I might as well get it right there.
True.
Well, it was kind of painful to get you to go to begin with.
I think I got the note what I got, but was it flu block, Amy? Do you remember? Or I
Yeah,
don't
we
remember.
got flu block.
So that's the baculo.
Yeah.
So we're
So-
What
probably
my pharmacist knew about were the difference between Moderna and Pfizer. Those were the only distinguishers.
Yeah,
But flu block would have the mismatched H3N2, right? Because that was formulated a year ago, and then the 2A2 clade arose after that. So
yeah.
I
And
don't
how
know.
maybe I'm not focused today, but are we absolutely positive that the glycans are required? Because like recently they've been spike has been deglycosylated, and there's a report that that's a better antigen actually.
Well, actually, as we go through this paper, I think one of my bottom lines was that the role of the glycosylation in all this is ambiguous. I think.
I agree. I think the amino acids are important.
I
Right?
think so,
yeah,
too.
so I'm not sure that I'm not sure that making it in eggs versus plants or versus baculovirus, I'm not sure that the distinguishing component is really the glycosylation. I
You
guess
recall-
is what I'm saying.
You will recall that biochemists, when they originally discovered glycosylation, referred to them as protein decorations.
Yes, I
They
do.
had no idea
I do know
of
that.
what they did.
Well, I do
Some
know
of them
that
are
for a
very-
fact
Some of them are dependent upon the terminal sugar in order to be taken up by a cell. I know that for some proteins, but for this one, I don't think that's true because you're injecting it already.
Yeah,
So-
but the
so-
other thing is you're absolutely correct that they just used to refer to them as decorations because
That's right.
my father
That's right.
has a friend from being a postdoc
Sure.
who has an entire company about glycosylation in California.
Oh, yeah.
And yeah,
But there's an antigen that trichinella contains, which is a glycan, and you could remove it. It's an O-glycan, and when you do that, then you only get the antigenicity of the protein, and that's the one thing we were looking for. Because otherwise, there's an array of proteins with the same glycan on it, and you get all of them reacted to when you don't remove it. So it was very important
So,
to know that
Amy,
we were dealing with it.
In response to your point, so the Scott Hensley, when he was on TWiV, he talked about a 2017 paper from his lab, which concludes that when you grow the vaccine in eggs, you get changes in the HA that alter glycosylation. And if you then say ferrets and humans exposed to the egg-adapted vaccine make antibodies that poorly neutralize circulating H3N2 viruses. And the paper it was focused on this idea that the glycosylation is what does that, although the current story is not clear that glycosylation has anything to do with it.
well,
So
I
I think-
just know that there was a paper that I saw yesterday in which they
Yeah.
deglycosylated spike and it made a better vaccine. I think it was like in Science Translational
No, I think that's-
Medicine or something or other, something else. I forget exactly.
That makes sense. The thing is, if you put a sugar on that shouldn't be there, it can alter the antigenicity. That's the point, I think. But you're right.
Well,
If you take
I mean, we're saying the same things, but
Yeah,
just
yeah.
from different points of view.
perspectives, yeah. So in this preprint, they say this 2A2 emerged early in the COVID pandemic. So that'll be early 2020, and then almost completely displaced other H3N2 clades throughout the world in 2021. And it has a number of substitutions in key epitopes on the HA. And they found, as I mentioned, vaccine effectiveness against this clade 2A2 was found to be low during a recent outbreak on a college campus in November, right? So there was already some hint in November. And now, of course, as Rich said, the MMWR says, yeah, there's a real problem. So this viruses, these H3N2 in the 2A2 clade, they have a substitution at amino acid 160, a threonine to an isoleucine that is expected to eliminate glycosylation motif at amino acids 158 to 160, which is part of antigenic site B. And they do some experiments to show that, in fact, yes, that change does eliminate a glycosylation site. And this is of concern because the glycosylation at that site can affect the binding of the HA to the receptor, which is sialic acid. So they did an experiment to address that. They have a panel of sialic acid derivatives of alpha-2,3 and alpha-2,6 linked. In other words, sialic acid linked to another sugar by different kinds of chemical bonds. And they find that the HA from this new virus does not bind strongly to many alpha-2,6 linked glycans. It bound poorly to most alpha-2,6 linked sialicide glycans, but had good binding to other kinds of sialicides. So there is a difference in that 2A2 clade in terms of sialic acid binding, although they say we don't know exactly what that does. They say the role for this glycan in shaping binding is suggested, but we're not for sure. But the real question is how does the biology of this new clade differ? So they do growth curves to ask whether the change in glycosylation has any effect. And they use primary human nasal epithelial cells, which is good. That's the right cell to do this in, not kidney cells. That's good. I'm glad they're using the right cells. They do growth curves, basically. And both 2A2 and the ancestor 2A1, they replicate very well. No differences in peak titers or the kinetics of production. They replicate well at 32 degrees, which is more like the temperature of the upper tract. So there's no fitness impacted by this change in the 2A2 clade.
So correct me if I'm misreading this, but there's two things going on here. There's a change in the amino acid sequence around this site. And,
158, right.
And because of that change, there's also a change in the glycosylation at that site, correct? On the hemagglutinin.
That's correct.
So the question becomes, as we've already said, how does this affect the properties? And then they have these glycan tests that you talked about. And it's, as you already mentioned, this variant does not bind a variety of different glycans, which are, this gets confusing because it's glycosylated, but it binds a glycan as well, okay?
Yeah.
But at any rate, the binding is changed. But then they go further and they say, okay, so if we take the original A1 and remove that particular glycosylation, or if we take the mutant A2 and add that glycosylation, how does that affect the binding? And neither of those things affects the binding all that much. The impression that I come away with is that the receptor binding or the sialicide binding that they're measuring has more to do with the amino acid sequence than it does with whether or not the thing is glycosylated. Is that fair?
I think that's correct.
That's correct.
Yeah.
But the other thing is just because you look at replication in the right cells, I'm not clear how that's related exactly to immune evasion or whatever they're calling it.
No, they were looking at fitness. They were just looking at fitness.
I mean, I
don't really. 2A2 has to be fit because it displaced 2A1, right?
Well, I'm not sure. Right. But so that's one way to look at fitness or one aspect of fitness, right? Mm-hmm. But there's a lot of other aspects of fitness, right?
I think what they wanted to know if this glycan change had any impact on fitness, if it made it better, I guess.
What is fitness? That's my question.
Well,
replication in the nasal cells is one.
It's when you work out 3 days a week at the gym.
Exactly. Do you lift weights or are you doing cardio?
That's right. Both. Walter.
Well, yeah.
You have to do both.
Well,
you can't do one without the other.
Fitness is the capacity of a virus to reproduce in the host, whether it's cells or in a population, right?
But reproduce how? So that's my question: is what aspect of reproduction? Because you're just basically looking at can it replicate itself, right? But in a host, there's a lot of other things that impact replication.
Yeah, sure. Well,
right?
they didn't do anything.
It stimulate as much interferon as the ancestral virus.
No, that's a good question.
So they could have
used ferrets.
They could have used ferrets. Exactly. They didn't do it. That would have been a good way.
Or they could have even assayed whether or not they could have assayed how much interferon they found in the supernatant of these infected nasal cells or whatever.
Yeah, that would have been good.
So just because they grew at the same rate in some cell line, I'm not sure. I'm not sure that you can say anything.
You can say they grow at the same rate. That's it.
In this particular cell line.
Yes.
Is this a plaquable virus?
Probably. Yeah. Most of the.
So you could look at plaque assays also, right?
You could. Yeah.
Well, I mean, again, plaque size. Are you going to look at plaque size? How does plaque size relate to fitness?
Number of viral particles that you get.
Yeah.
Well, each plaque isn't one viral particle, right? But you can have large plaques, small plaques, medium plaques, fat plaques, small, skinny plaques, blah, 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 blah.
Skinny plaques. Is that from working out?
Yeah, that's from, they only do cardio. They don't do any muscle toning.
I think their focus was really on antigenicity, which.
Well, I'm not sure that you can say anything from antigenicity from just growing it in nasal cells, right?
No. But the next set of experiments addresses that. So let's talk about that.
So I do want to do an aside here with these cells because I've become fascinated by where people get these specialized cells and how specialized they actually are, because they call these primary nasal epithelial cells, and they give a reference to a company called PromoCell that sells these for you to you, and these are genuinely, as far as I can tell, they say normal human nasal mucosa. Okay, that they will sell you 500,000 cells for $772, and then if you look through the methods for culturing these, they do them on transwell inserts, so they're appropriately oriented on a membrane, and it takes them a month to differentiate them appropriately in these and do the experiments. So this is no joke.
So, Rich, do you think
I think the motivation here is to get some sort of a cell culture that is as close as you can possibly get to a normal respiratory epithelium.
Do you think they're getting these cells from dermatologists?
I have no idea. I don't know. They talk about donors, and in fact, if you look at this thing, you can fill out a special request to get HLA typed cells. So you can get cells from HLA donors with specific HLA types. It's incredible how specialized this is.
Isn't this what Anne and Jim do, Vincent? Is that they have donors for their Rhino C stuff? They get donors and they
believe so, yeah.
Yeah, they take healthy tissue, like from, I don't know, from people who have lung biopsies or something or other, right?
Didn't you do some primary respiratory cultures? Or was that from mice?
I made them from mice.
It still took a month for them to grow, right?
Oh, yeah, easily.
Yeah. But at least you don't have to pay $700 for those.
Well, no, but like when we did the IPS, when we bought the terminally differentiated neurons, if we bought that I don't even think we got 500,000 cells for $750.
Expensive.
Like we got more like 150,000 cells for like that amount.
And
This is
the
like-
medium is very expensive also, right?
Yeah, because you have a ton of different, very specific growth factors.
Yeah. It's
Yeah.
extremely expensive. I think we estimated to do some experiments was going to cost $30,000 if that number pops into my
Ouch.
head.
Yeah, that's about right. And then if we did it ourselves where we made the cultures, like from iPS cells, it was like an extra 50.
Yeah, we were going to collaborate with someone. He said, okay, we can do this with you, but it costs $30,000. It's like, yeah, I've got 30,000. Amy has 30,000 just to try something. No.
Right, which is why at the end of the day, I walked down the hall to his colleague and did it for mice. And they I mean, it was much cheaper and everything. Yeah.
All right. So they want to know if, what's the, is this 2A2 clade virus antigenically distinct from the vaccine component? All right. So they do neutralization assays. They have serum collected from 40 individuals before and after getting the vaccine, the quadrivalent vaccine, Flulaval. I guess it's made in Canada, right? Flulaval. And they tested neutralization of wild-type H3N2, egg-adapted 2A1, which is the virus in the vaccine, and then the circulating 2A2 virus. Okay? So, and 2A1 has amino acids, asparagine, tyrosine, and threonine at positions 158, 159, and 160. And that gives you a glycosylation site. That happened from pathogen eggs. Egg-adapted 2A, that's the wild type. Egg-adapted 2A1 has asparagine, tyrosine, and lysine at the 3 amino acids. So it's a threonine to lysine. And that gets rid of the glycosylation site. So egg adaptation gets rid of the glycosylation site. All right. So what's the result? Most individuals had antibodies that neutralized the egg-adapted 2A1 strain. That's the vaccine strain. That makes sense because they were vaccinated with it, right? But not the wild-type version of the strain or the 2A2 strain. Antibody titers were reduced at least tenfold comparing 2A2 to 2A1. And they say that's probably an underestimate. So basically, and we saw this in an MMWR report in ferrets, right? The same thing. Antibodies induced by the vaccine, which has the 2A1 virus, H3N2, poorly neutralize the 2A2 clade virus. So that's why it's doing so well in humans this year. They said we were hopeful that the egg-adapted H3N2 would be closely matched to 2A2, but it wasn't. They're mismatched. And they say probably because of amino acid differences at 159 and 160. They don't really know what the glycosylation is doing.
Vincent, when are we going to see an RNA virus roll us?
An RNA vaccine?
Yep.
I think it's in testing. Didn't you tell me, Amy, that Moderna's doing a combined SARS-CoV-2 vaccine?
Yeah, they're doing SARS-CoV-2, influenza, and RSV.
But Dixon, it's not
I
going
think
to.
actually there might have been a fourth one, but I forget what it was.
Yeah.
Dixon,
have to
you're still going to have to pick the HA, right?
Well, that's the point. Didn't they also identify a universal antigenic site that had nothing to do with HRA?
Well, they have on the stem of the HA, there are conserved epitopes that do not apparently vary.
Right.
Yeah,
And
but you don't get a really good neutralization. You don't really get good neutralizing antibodies against that. Some of the tests are flawed that they use to do that.
I thought they were looking at other parts of the virus besides the H or the N.
I don't think
Well,
so.
the HA and the NA are the ones that can give you neutralizing antibodies, right?
Yeah, but
Well, if
remember
you
what
want
you
to
said
go
about
that
that.
way,
I mean,
we've always
maybe
been critical of just neutralizing antibodies.
Yeah, I agree.
The other protective mechanisms involved that you're overlooking,
for
like T cells.
For flu,
Yeah.
Dixon,
Well,
these vaccines do not give you good T cell responses,
okay.
and maybe that makes them so dependent on neutralizing antibodies. And if you have a mismatch, you're screwed. So maybe the key would be to make a vaccine that gives you better T cell responses, and an mRNA vaccine could do that. Maybe, yeah.
Or maybe you can use something like Matrix as a T cell, as a basis of a T cell vaccine.
I think that's a good idea. I think that's a good idea. Yeah. So, we need to do better because 16% is just unacceptable. And I think it leads to people saying, I'm not getting this vaccine.
Well, that was my next question was, as we discuss this, how many people now are going to, how many people have we now led to be discouraged to getting their annual flu vaccine? We've not really advocated. It would be hard to advocate from this conversation for getting the vaccine.
But first of all, it's April, so nobody's going to get vaccinated anymore here in the Northern Hemisphere. But in this preprint, just as in the MMWR, they say studies have shown that even when there's a mismatch, you still get protection against severe disease
it sounds
and death.
like the same argument for COVID.
I was just about to say, it's just a recapitulation of that same argument. And it comes down to what group you're in and how many people actually get severe disease between the vaccinated and the unvaccinated.
So we're still waiting for those results?
3% difference? Is it 1.5% difference? Is it 10% difference?
Are we still waiting for those results?
Probably.
What results?
Of this year's experience with the flu vaccine versus the death rate from flu.
Yeah, but there haven't been any studies this year. No. By the way, in this MMWR, they say the percentage of these patients who got vaccinated ranged from 31 to 64%. So that's the percent of patients who got the vaccine, which is kind of like COVID is maximum 64%, right?
I mean, yeah, but that's kind of poor at 31%.
Well, 31 is horrible. And part of it is because people think flu is not a big deal and the vaccine doesn't work.
Well, I think it's more that the vaccine doesn't work.
It's not a great vaccine. I agree.
The other thing they're used to, though, is the annual death rate. That doesn't startle anybody anymore, so they don't take it as seriously because it's not a newly invented virus or it's not a newly introduced virus. It's something we can live with this. Yeah, that's okay. It only infects older people.
Yeah, I know.
No problem.
I mean, we can have 40,000 deaths a year in the U.S. due to flu, and it doesn't faze people. You're right.
Correct. It should, but it doesn't.
I get it. All of us get flu vaccine every year, right?
Yep.
And why is that?
Because we don't want to die from it.
Unless I go with you.
I do not want to die from flu.
Well, now you're going to be gone, so I can't. I have no excuse, right?
I'm still going to have to come and take you.
There are too many people out there with tragedies in their own families where they didn't vaccinate younger children, and because of that, they lost children to the flu vaccine, to the flu virus, rather. I know one in particular, but there are many, many, many others like that.
So, Dixon, when people have that experience, do they then get vaccinated?
Yeah, they do.
Yeah.
But it's too late. I mean, that's a horrible lesson to learn.
It's absolutely right.
I mean, that's a lesson you should never have to learn. You should be smart enough not to do that. But on the other hand, I have relatives who are older who didn't get flu vaccines and only because their significant other was even older than they were. And they got severe flu and developed pneumonia. Did they then follow up with getting vaccine?
So, Amy, you use that word, they're not smart enough. I beg to differ with you. These are smart people that are trying to outthink this whole system. And they find excuses. And they find the anti-vaxxers are not uneducated. They've often come from PhD backgrounds, et cetera. So,
well, what she means is they're not smart enough to get vaccinated.
The word smart, maybe cautious enough, or maybe...
All right, it was a bad word.
I use a different word. I wouldn't use smart.
Okay, it was a bad choice. I agree.
What would you use, Dixon? That's a good point. What would you use? They're not...
Arrow on the side of caution.
How about they have too much hubris?
They're trying to outthink this thing in a way that allows them to call themselves part of a group. And they resonate with that group. The anti-vaxxer group has a lot of other issues besides vaccination.
One of their arguments is, I don't want to take too many vaccines. That could be a problem. Did you hear about this guy who got 90 shots of COVID vaccines in Germany?
No.
No?
Good
Did you hear
Lord.
about this, Rich?
Yeah.
What happened to him?
Oh, he's fine. He just wanted to sell the vaccine passports.
Did he dehydrate?
No, each time he got a vaccine, he got a new card and he sold them. It'd be interesting to follow up to see what happens to him. He could be the poster child for.
Right.
Well, it's just one person.
Right.
All right. Anyway, so there is a mismatch this year. Not good. So this year we're going to put 2A2, H3N2 into the vaccine, and we'll see what happens. Stay tuned. It's a page turner, folks.
It seems to me that based on some of this stuff, when they do these surveys to figure out vaccine effectiveness, they kind of need to sort people out as to what vaccine they had. Because, you might get a different result with the egg-adapted vaccine or the, flu block or one of the others that wasn't grown on eggs.
Yeah, that's a good point. But of course, they're all a mismatch, right?
Yeah.
So the virus is a mismatch, but whether it's glycosylated or not, that could make a difference. Yeah.
Have we ever actually nailed this vaccine?
Yeah, we do sometimes.
It was absolutely perfect. H5N1, for instance. Or
there's no H5 in people, but we don't have it in the vaccine anyway. But yes, there are years when there's a good match, for sure.
And then the death rate goes down.
Yes, and the death rate can vary from a few thousand to 40,000. That's the historical range.
Right. So there's historical evidence for maintaining this high level of vaccination.
Mm-hmm.
That's what I would point to.
Well, the problem is, Dixon, many people never get flu vaccine and then never get flu. And they say there's an argument. So it's hard.
No, you're right.
But the one time they get it and die, then that's it. It's,
it's too late. That's the point where it's too late. That's right.
Flu-Laval, by the way, is a GlaxoSmithKline product. I looked that up.
And what is it made in? What kind of?
I don't know. It's quadrivalent. I haven't looked into it carefully enough. I'd have to. Actually, what I do when I want to know, let me even do this. When I want to know the details of a vaccine nowadays, is
package insert,
get
right?
the package insert.
Yeah.
You can keep on going. I will see if I can.
All right.
That's a Swiss company?
Glaxo.
Swiss.
Here, I've got Smith?
It's Swiss?
I think
it is. British.
Yeah, I thought GSK was British.
I know. Okay. There's so many corporate piggybackings that is hard to keep up with the main offices.
We have another paper, which is really quite provocative. It's a Nature article. FC gamma receptor-mediated SARS-CoV-2 infection of monocytes activates inflammation. This comes from a group of people from mostly Boston. There's one group from the Oswaldo Cruz Foundation in Brazil. There's one group in University College London, but mostly from Harvard and the hospitals in Harvard and so forth. And the first three authors are Caroline Junqueira, Angela Crespo, and Shaheen Rajbar. And the two corresponding authors are Michael Philbin and Judy Lieberman. As you know, in some patients, you can get severe COVID, which is characterized with increased inflammation. And they use a word that I hadn't heard, inflammaging, increased chronic inflammation associated with aging. I'd never heard of that. Inflammaging.
That's a new one to me.
And it's, of course, important to understand the basis for this. And in part, we've talked a little bit about this lately. We've talked about it in terms of response to the mRNA vaccines. But in infection, in a patient who is infected, the infection is sensed, and sensors like inflammasomes are activated, which then drive the synthesis of cytokines, including IL-1, which they call arguably the most potent inflammatory mediators. In addition, NF-kappa B, TNF, Th17 cytokines are also induced, and they can cause severe inflammation as well. Now, the inflammasome, and we talked about this last week when we talked about IL-1 and mRNA vaccine inflammation. Inflammasomes can sense a molecular pattern. They then recruit an adapter protein called ASC. That's what adapter molecule apoptosis associated speck-like protein containing a card. That's what ASC stands for. But for the purposes of our discussion, when the inflammasome detects something foreign, they recruit the ASC adapter. They assemble into larger complexes that then activate caspase-1, which processes pro-cytokines as well as a pore-forming protein called GSDMD. Would you like to know the meaning of GS?
Absolutely.
Gasdermin. It's gasdermin. All right, let me look it up again because I don't remember. GSD. I know it has gasdermin in it.
Gasdermin D. It's in
gasdermin
the abstract.
D.
It's in the abstract.
Gasdermin D. And then gasdermin D has pore-forming properties. It disrupts the cell membrane, the cell dies, and then if there are any cytokines in the cell, they're released, right?
Right.
It's called pyroptotic cell membrane rupture.
So this fascinates me. These cells, macrophages, right?
Yeah.
Pick up, and this is critical. I won't spoil it, but they somehow get infected, okay? And they have this sort of innate inflammatory response. They fill up with all of these things that are inflammation signals, and then they die basically in order to release this trove, right? It's amazing.
Right. And normally this trove is beneficial, right? Because the beneficial recruits immune cells and those help to clear the infection and so forth. And lactate dehydrogenase is one of the things that is released. It's an enzyme that is released from these dying cells, from these pyroptotic cell membrane rupture. And it is an indicator of pyroptosis and cell death. And then they say it's one of the best correlates of severe COVID. LDH levels, you can measure that in the blood of patients. So this paper is trying to get at the mechanism here. What's going on? So first set of experiments, they take blood from SARS-CoV-2 patients and they look for markers of inflammasome activation and pyroptosis, these processes that I've told you about just now. They take mononuclear cells. They have healthy donors, HD, and 22 COVID patients in the emergency department. And they are stained for various markers and also with a chemical called zombie yellow that will enter a cell that has a damaged membrane. So that's, an indicator of apoptosis that this molecule can get in. Isn't that just great, zombie yellow? Don't you love it? Or am I just...
No, I like zombie yellow.
Zombie yellow is just cool. All right. 6% of monocytes in the COVID patients took up the zombie dye, which would be an indicator of pyroptosis, right? This membrane breaking process that we have discussed. And they could ask what kind of monocytes these are by doing flow cytometry and measuring surface markers. They turned out to be classical monocytes. And they find lower numbers of these cells in these patients as well, which suggests that something's going on to them. These monocytes they can stain them for nucleocapsid protein. And so they're saying somehow the virus must have gotten in because nucleocapsid protein is inside these monocytes. And the nucleocapsid is only in monocytes that produce Fc gamma R3A, which is an Fc receptor. It will bind to the Fc portion of antibodies. And that particular Fc receptor, also called CD16, is known to be a mediator of antibody-dependent phagocytosis. So in other words, when an antibody molecule binds, it could be a virus or something else, monocytes can bind the Fc portion via this receptor and bring it into the cell for destruction. That's the normal course of events. Now, they also looked at markers for inflammation. All inflammation markers that weren't specific for pyroptosis were elevated in the COVID-19 plasma. But they did not detect interferons above baseline. All the pyroptosis markers were elevated in the COVID plasma compared to the healthy donors. So this is, Amy, what you had said before. They should have looked for interferon. So they're not seeing interferon in these patients, which is probably part of the reason they get so sick, right?
Yeah, it's known that it's not supposed to be an interferon stimulator. But the other thing is they look at beta and gamma. Where are they taking these samples from? Because the virus is a mucosal.
These are blood samples.
Yeah, these are blood samples.
So they should be looking. They should be looking at for lambda.
Okay.
Because it's a mucosal surface,
right? Yeah,
and
but of
so
course
the mucosal surface is protected by lambda, not beta or gamma.
Yeah.
I mean, gamma, I guess, is the indicator for T cell activation.
All right. So then they ask, are these markers of pyroptosis, do they correlate with severity of disease? And the answer is yes. They find higher levels of these markers in patients with severe disease.
Mm-hmm.
All right. So far we have this suggestion that the monocytes might be dying of pyroptosis, and they're releasing cytokines, so maybe that contributes to poor outcome. So they ask, if we take monocytes from patients, can we do this? Can we get them to undergo pyroptosis? So they isolate monocytes from patients with different disease severity, and they look at markers of inflammation, the inflammasome adapter, the gasdermin, and so forth. And here's another thing we have to introduce. When the inflammasome becomes activated, they form big inclusions in the cytoplasm that are called specks, because they have ASC and caspase in them, and you can detect those with specific antibodies. So they're called ASC specks. 4% of monocytes from COVID patients and 1% from non-COVID patients, none of the healthy donor samples had specks, caspase and ASC specks. Okay, 4% monocytes from COVID patients, 1% from non-COVID. And so basically they're thinking, these non-COVIDs have other respiratory diseases, so maybe those infections are stimulating this process in the monocytes, but it's more extensive in SARS-CoV-2 infection.
So this is apparently not a new technology, but it was new to me. Imaging flow cytometry, which they not only do you stain cells with fluorescent markers and sort them to individual cells, but take pictures of individual cells. And this machine actually sorts out the pictures with different fluorophores of the individual cells, which just absolutely blows my mind.
Yes,
and
that's pretty cool.
And that allows you to distinguish this speckling phenotype.
That's right.
From the more uniform distribution. And it's striking. It's amazing. I also, so maybe you already said this, but I mean, one of the introductory things for me was the monocytes are apparently, going into this naively, going back a little bit, monocytes are apparently taking up virus, but they don't have appropriate receptors. They ought to be resistant to infection.
That's right.
And so all of this stuff with the antibody-dependent uptake is the mechanism by which the virus gets in. And it's a funny kind of thing because that's all evolved for the purpose of getting rid of the virus.
That's right.
But in fact, the virus, as we'll see, replicates sufficiently to set off this whole cascade.
Yep. Yeah, but some things are going off here, as we'll see, right?
Yeah, Vincent, you mentioned early on that LDH was part of this
So
picture.
LDH is one of the proteins released from these pyroptotic cells, right?
Now, that's an anaerobic process, right?
Lactate dehydrogenase.
Yep.
But it's
You
just
usually get that after the oxygen is all used up. Because that's what you get accumulating in your tissues when you work out, Amy, too much, and you stress out your muscles. All the glucose is converted to LDH pathways, and it's an anaerobic process. And that's why it hurts when you go home.
Yeah, but it's also a marker for
No, I understand that. So I'm asking you a fundamental question, whether the inflammasomes are anaerobic.
No, I don't believe they are. I mean, lactate goes lactate to pyruvate and back, right?
Yeah, but I also thought that there were some early COVID treatments where they were supersaturating the patient with oxygen, and that's why maybe that was trying to address this without knowing actually what was going on.
No, I think that was an oxygen deprivation issue there, right? I mean, lactate dehydrogenase is in many tissues because it's part of the glycolysis, right? So I think
it's part of the anaerobic glycolysis. It's not aerobic; it's anaerobic. I know that for sure.
But it's
a marker used for a lot of things.
As soon as the myoglobin is used up for the oxygen in your muscles, it converts to lactic acid.
You're absolutely right. It does it in the absence of oxygen. That's absolutely right.
Yeah, but it's used as a marker for lots of things. I mean, one of my
I
parents
understand that.
Who has
but it's
cancer,
interesting that it should pop up here.
Well, I mean, it's cell death, right?
Yeah, it's cell death. It's a marker for cell death, and in this case, it's a specific. It's a specific kind of cell death. But I don't think the anaerobic aspect is really playing a role here, Dixon. Okay.
Yeah.
No, I'm a little confused about why it's confusing to find virus in monocytes. I mean, it's non-specific phagocytosis.
Yeah.
It's sampling things. So,
right.
I mean, you're going to have, I would assume that if you randomly pulled out 100 monocytes, one of them is going to have be nucleic capsid positive just because it's a random sampling of the environment. So I'm not clear that I'm not clear that I would call that infection.
No, it's uptake.
Right.
Right.
But Amy, in the absence of antibodies, would monocytes still take up virus particles? They probably would, right?
Yeah.
Yeah, periodically, depending on how much is concentrated, what the local concentration is of the environment that it is present in. But the antibody seems to be like a honing signal, right?
Yes, sure. I think it increases the efficiency. But you can imagine if a monocyte goes to an epithelial barrier that's infected and releasing lots of particles, it could take up.
Right, but there I would assume, if we take the analogy of 1 to 100 for this, I would assume it's 1 to 10,000 for that process. So it's much rarer, much harder to find. But I think that we know that this idea of leukocytes being infected has been previously discussed, where I kind of was like, because it's non-specific phagocytosis, right?
Yeah.
That's right. That's not
if
infection.
That's true, then everybody with SARS-CoV-2 should have some infected monocytes. And this paper says only 6% of the blood monocytes.
Of what you could find, of what you could sample, right?
Well, the sensitivity of the test is such. That's right.
But also, there's something going on in infected patients, which we'll get to.
Right. I mean, I would assume that if I looked for rhinovirus, I would find 3% of I could find 3% of monocytes infected with rhinovirus when you had a cold.
Well, these 1% of non-COVID respiratory patients have specs. So other viruses are causing specs, but it's, there's something different in the COVID patients. Let's get to that. Anyway, all these experiments they look at the monocytes, with these ASC specs, their plasma membranes are screwed up. The gas dermin is in the cell membrane. Zombie dye is. So basically, they conclude that monocytes are clearly undergoing pyroptosis. Okay. Then they want to say, okay, there are many inflammasomes. They're like, what are there? 25 different inflammasomes. Which one is involved here? And they use a variety of approaches, including confocal microscopy, and they seem to say that this is one called NLRP3. So inflammasome specs, gas-dermin membrane localization and cleavage, dying annexin, zombie-positive monocytes, IL-1 cytokines being produced. All of that shows that the COVID-19 monocytes are dying of pyroptosis. Okay, so what is activating the inflammasome in these monocytes? You probably have already guessed, but they say, okay, it's probably infection, right? But monocytes don't have ACE2, as we've already said. They don't have ACE2. And then they say, well, they do have CD147, which has been proposed to be another receptor, but they say this finding is controversial, which I think is a nice way of saying we don't actually believe it. But monocytes do produce 3 FC receptors, FC gamma receptors. And these are increased in COVID patients. So these could recognize antibodies, right? And pull the virus in. If the antibody is bound to the virus, it could pull the virus into the cells. And you can find spike antibodies early in infection, about the time when patients develop inflammatory systems. And that's the case in their cohort. They have antibodies that can bind virus. So they did some staining. They took these monocytes from the patients and co-stained, this is healthy donors and COVID patients, for nucleocapsid protein, double-stranded RNA, and ASC, that protein that's recruited to the inflammasome. So N, of course, would be the presence of virus particles or virus proteins in the cell. The RNA reproduction, replication of the genome, and the ASC would be the pyroptosis. So the healthy donor monocytes don't stain for any of these. 10% of COVID monocytes stained for N or double-stranded RNA, and 95% of the N-positive monocytes also have viral RNA in it, so they think there's viral replication. And all these cells, and RNA-positive cells, have ASC specs. So they think that the inflammasome activation and the subsequent pyroptosis is all triggered by infection of monocytes by SARS-CoV-2.
Well, it's not infection, it's uptake.
Yes. So just to clarify, what would you say is an infection? The whole cycle?
Yeah.
From beginning to, okay.
Well,
I would agree.
yeah, but it's specific, right? An infection is initiated by receptor binding, right?
Okay, that's fair. Okay, so the respiratory tract, of course, is the main infection site of SARS-CoV-2. So they looked at macrophages in lung autopsies, whether they were infected with SARS-CoV-2 and whether they had activated inflammasomes. Because so far we're taking them out of the blood, right? Which is okay, but that's not where the active infection is going on. And in fact, the lung macrophages have activated in ASC-containing inflammasomes. But interestingly, the epithelial cells and the endothelial cells in the lung from the same patients don't have activated inflammasomes in them. And what's interesting, this is an interesting observation. So a fraction of cells, the monocytes that have specs, only 8% of them were nucleocapsid positive. So the others are not exposed. What is the word you used, Amy? Exposed instead of infected?
They have not taken up.
They have not taken up.
They have not taken up virus.
Which
and they say maybe
that it's some kind of schmutz that's being released.
Schmutz released from the dead cells, right? They're being taken up, and those are activating those cells in the absence of uptake of virus particles, right?
Zombie goop.
Well, to me, there's a certain fraction of these cells that are activated because they have taken up SARS-CoV-2, and there's a certain fraction of these cells that are activated because they're busy with other things.
Right. Okay.
There's
Or
all sorts of
the ones that are activated by taking up SARS-CoV-2 are doing something, releasing some secondary signal molecule that then
Yeah,
signals
that's right.
to the neighbor, which I think is the point of the inflammasome, is that it forms a pore that you release, I don't know, some death signal to your neighbor.
So some of those macrophages are taking up debris from dead and dying macrophages.
Right. Yeah. Correct.
So that's like a secondary effect.
Exactly. That's exactly. Or other cells. Yeah. Other dead and dying cells.
Exactly.
Right.
Well, isn't that what I just said? Some secondary signal?
Yeah.
It is.
Dixon's just making sure he's got it. That's all.
That's exactly
I'm just
right.
making sure I've
Just
got
like
it
Rich.
too. Sometimes.
Let me get this straight. Rich says, let me get this straight now. Correct me if I'm wrong. I was at a meeting once in which one guy at the speaker's podium pointed to a colleague of his in the audience. He says, now I'm going to say something here. Correct me if I'm right.
That's good. I like that. All right.
Now we have, and you didn't know why.
Now we have a series of experiments that are very cool. Okay. First, they say, can we infect monocytes in culture, right? So they have SARS-CoV-2 where they've stuck in a fluorescent reporter called neon green. Man, this is great. We have zombie yellow. Now we have neon green. And then they have monocytes from patients. So they take them alone or they prime them with lipopolysaccharide, which will activate them, right? And then they infect them with SARS-CoV-2, and then they have an IgG1 isotype antibody that is known to bind FC receptors on the cells, and then they have anti-spike antibodies. There's one that's non-neutralizing and one that's neutralizing, and then they also have pooled serum or pooled plasma from either healthy donors or COVID patients. So they're all going to see if these facilitate infection of monocytes, right? And the plasma, they do heating or not, because heating will destroy complement if complement's involved. Okay? So they do this infection with or without the antibodies. And then 48 hours later, they do flow cytometry to look for nucleocapsid protein, double-stranded RNA, and ASC. Okay? So what are the results? Without LPS, very few, without LPS, anti-spike antibody, or pooled plasma, very few monocytes take up the virus. But if you add anti-spike antibodies or COVID plasma, you increase infection of the monocytes.
Uptake.
And the uptake, sorry, thank you. It's okay. I'm going to miss it for a while. If you heat the serum, it doesn't matter. It's still, the monocytes will still take up virus, so it's not complement. If you deplete the plasma for IgG, then the monocytes don't take up virus. What's cool, I thought they deplete for IgA, the plasma, there's no effect. So IgA is not involved, and that's because IgA doesn't bind FC receptors, right?
Right.
So this is telling us that the uptake of SARS-CoV-2 by monocytes is mediated by spike antibodies binding to the virus and binding to the FC receptors on the monocytes. I thought those were a nice series of experiments.
Yeah. It's good.
And they go on to look at a variety of isolates of SARS-CoV-2 that have different affinities for ACE2. They say this is quite clear that this is all ACE2 independent. And I think there's no doubt about that. So then they do an experiment to address whether disease severity or antibodies induced by vaccination are involved in this uptake. Hey, Amy, there they say monocyte virus uptake.
Yeah, it's good.
They listened to you. They switched.
It's good. Yeah, so infection, because if there's no difference, then you negate the idea that there's a cell tropism for anything.
Yeah. So now they're using serum from COVID patients or from mRNA vaccine recipients. And the serum from patients with mild disease or severe disease. disease severity doesn't affect uptake. They're both similarly allows virus uptake. But the vaccination plasma did not facilitate uptake. I think that's a really important result.
It's very cool.
So it's only infection with virus that's leading to this. Vaccination is not causing this problem, so get vaccinated.
Well, the other thing it says is that there must be a different IgG, right? You're inducing two different types of IgG.
In what?
So, like, we measure IgG to determine whether or not you have neutralizing antibodies against the vaccine, right?
Yeah.
And now you're telling me that those IgG antibodies don't allow the monocyte to uptake virus.
So there must be vaccination. Yes,
right.
absolutely.
So there must be one must be like IgG1 and one must be like IgG5 or something.
They think it has to do with fucosylation of the FC.
Well, why would that could be? But why would the FC?
I don't know. But they say severe COVID patients have antiviral IgGs that are afucosylated in their FC region, so less fucose, which is a sugar. And they bind better to CD16, one of the FC receptors. I don't know why patients would have that, right?
Why would you change that?
That's an open question. Somehow, there's a difference between infection and vaccination that results in a difference in the level of fucosylation, and that affects this whole thing. And that's this goes all the way back because they talk about. Antibody-dependent enhancement, which was a concern early on that if you vaccinate people, perhaps you would get enhanced disease because of their immune response. And what they're saying is that
it's the other way around.
It's the other way. Yes, exactly. It's the other way around. The infection through this route has the potential to give you enhanced disease. Perhaps partly dependent on the fucosylation state of the antibody. Vaccination doesn't do the same thing because the antibodies that you get when you're vaccinated are appropriately fucosylated.
What did the lady from Peter's lab who's at Stanford? You talked about her paper, and she has a series of papers, and she wrote to you about discussing her paper or something or other on TWiV. So what's her story? Because she has a lot of stuff about the different fusilation thingies.
Yeah, that was Tanya Wong, right?
If I could remember her name, I wouldn't have said the girl from Palazi's lab.
Didn't we?
But you talked about
fucosylation. We did a paper from somebody studying this on a previous TWiV. But I forget where it was.
I think we did her paper, which said that patients have decreased fucosylation of the FC receptor.
So this would be consistent with that.
Yeah.
I think she has a series of papers.
Let's see here. Okay. So here in December 2020.
So long ago, I can't remember.
'Twas COVID in the aerosols. We talked about association of afucosylated antiviral IgG with severe COVID. And that is whose paper? That's a science paper, and that is Tanya Wong. Yeah. So I think, no, she's not on that paper, but I think that was her story for another virus, Amy, that I don't recall.
I'm sure. I think that was her thing. She's from Ravich's lab and then went to Peter's lab and then went to Stanford.
And Ravich is an FC receptor guy, just so that people know. Anyway, they have serum with low and high afucosylation. So they have 8%. It's hard to wrap my head around. Low or high afucosylation, right? So high afucosylation would be low fucose, right?
Seemingly.
Anyway, low or high afucosylation. Low afucosylated IgG did not increase infection. In other words, it has a lot of fucose on it. High fucosylated high uptake. Yeah, and high fucosylated did stimulate uptake. So I think that's a key issue. Why does infection but not vaccination cause a difference in fucosylation, right?
Yeah, that's what I want to know.
I would like to know that.
And curious minds want to know.
So what is the FC receptor leading to uptake? They incubate monocytes with their virus in the presence of serum that either was depleted or not of IgG, and which in addition has antibodies to potential receptors. And what they find is that the uptake is mostly mediated by two FC receptors called CD16 and CD64. Not ACE2, not CD147, et cetera. They also said, this CD16 is on other cells. It's on neutrophils. It's on cytotoxic T cells. It's on NK cells. So could the virus enter those by a similar mechanism? They didn't see any increased cell death in lymphocytes. They say, so we didn't study them further. No uptake into lymphocytes. Our neutrophils, what is the bottom line? Infection was not detected. Uptake was not detected in patient neutrophils. Okay, that's the bottom line there. So it seems to be mostly monocytes. And the last section is, okay, the virus is being taken up, but do the monocytes make infectious virus? In which case we could call it infection, as Amy would say, right? So they use an antiviral, remdesivir, which inhibits the RNA polymerase, and they use camostat mesylate, which inhibits TMPRSS2 on the plasma membrane, which cleaves the spike for entry, right? Monocyte uptake was unaffected by camostat, but inhibited by remdesivir. So it looks like the end signal, that's what they're looking for, goes away when you put remdesivir. So the suggestion is that the RNA is being reproduced. And in fact, they can detect subgenomic RNAs in these cells. It looks like the virus is getting in and beginning to replicate its genome. Do they make infectious virus? They did a plaque assay. And, interestingly, they say infectious virus is detected in patients' plasma only with very sensitive assays. We did not detect virus by plaque assay in 9 plasma samples. And so, this idea that there's viremia, I don't think it's actually virus. I think it's RNA-emia, right?
Yeah. But I think actually this set of experiments caused Ben Tenuver to tweet that they had only done RT and it was insufficient. So clearly they did plaque assays.
No, they did plaque assays.
Yeah.
Ben is the guy who does only PCR. He doesn't do plaque assays.
I know. I thought it was a bit strange that he tweeted how they hadn't done the right experiment.
Why would he say that? They did the right experiment.
I guess
this is the problem. This is the problem with Twitter.
I don't know. I didn't ask him.
You didn't have a Twitter argument with him?
No, I don't. I don't have any Twitter arguments.
You don't, and you still have 1 million Twitter followers.
I don't even tweet. I don't tweet. No, but
anyway,
yeah.
They allow monocytes to take up virus, and then they look at the culture supernatants 48 hours later. There's no infectious virus. Yet if they do the same with vero cells, they can easily detect it. So they say monocyte infection did not produce infectious virus. And I would add, Rich Condit would say at the limit of detection of your plaque assay, right?
Right.
But it's probably not very much, if any.
Well, if you're making less than 100 PFU.
Yeah.
Because that's basically the limit of detection. Undiluted, if you put 100 microliters of undiluted, you're at -2, which is 10 plaques. 100 plaques, I mean.
And they call this an abortive infection, which is correct. It seems to get in, make a little RNA, some proteins, and you don't make infectious virus. So I would say these monocytes are non-permissive. Would that be acceptable?
Yes.
So this is very cool. And I think it's a very neat story. So this may account for severe inflammation in COVID patients, this uptake of virus into monocytes.
They account for part of it, not the
part
full
of it.
mechanism.
Well, they actually say that it's probably a major contributor to pathogenesis, but I don't know how you can say that because it's probably multifactorial, right?
Yeah, that's what I don't
Now
understand.
they say, this is cool. They say, why hasn't this been widely recognized? And they say, well, there are three reasons. First, many people use frozen cells that they thaw, and these cells don't survive. So you need live cells for this kind of uptake.
Coming from the man who thought you could freeze a spleen.
My impression was that it is specifically the activated cells.
That's right.
That die. So you select against the ones you would really like to see.
Yeah.
And they also say few people have looked for monocyte uptake because monocytes do not produce ACE2. And this is this focus on ACE2, right? That it makes people look the wrong way.
Well, they obviously didn't read the Diamond paper.
That's right. And they also say Fc gamma mediated uptake of antibody-coated virus into monocytes is a double-edged sword. It's exactly what we were saying earlier. It's a defensive mechanism, but it can also lead to the monocytes getting broken open and releasing all these mediators. And they say this is cool the way they say it. The inflammatory mediators spewed out from pyroptotic monocytes can cause a cytokine storm. Isn't that great? Spewed out. Nature let them keep spewed out.
Very visual.
All right. So what can we do about this? Those of you who are listening and you're still with us, which probably there are 5 people left. What can we do? So this knowledge has to be actionable, right? So they say, do we have drugs that could, inhibit inflammatory cytokines or even Gas-dermin? There are, but they say clinical trials that evaluate antibodies that inhibit inflammatory cytokines. Anti-IL-1 beta is called canakinumab, and anakinra is anti-IL-1RA, and also antibodies to IL-6 and IL-6 receptor. They say have shown at best weak protection, probably because of suboptimal timing or because any one cytokine is one of many mediators. And that's why dexamethasone works, because it's a broad sword, right? But they do say that 2 FDA-approved inhibitors of Gas-dermin and 2 approved inhibitors of Gas-dermin, disulfiram and dimethylfumarate, are currently being evaluated in clinical studies. And they have proven beneficial in mouse models of sepsis, which they say has overlapping features with severe COVID. So that's cool if some of those work. All right. Finally, this is antibody-dependent enhancement. There's no question about it. But they say plasma from vaccinated individuals did not promote monocyte uptake. Therefore, ADE is not a concern with respect to vaccination. So many people are worried about ADE. It's not a concern. And what about afucosylated antibodies? They don't go up after the afucosylation is not a feature of vaccination. It's a feature of infection, which we've mentioned before. And so we need to understand how these features, this afucosylation, leads to a difference in FC receptor binding. So overall, I think there's a lot of really good results in this paper. I would say this one has a high quotient. What is that funny quotient we used to use to say how much data was in a paper? Oh, I can't remember now. Do you guys know what I'm talking about?
No.
It was some funny term we used when a paper had a lot of data in it. This paper has a lot of data, and it's really well done.
I really love the pictures.
Pictures are
and
great.
How to see so dramatically how the components get reoriented in the cells and how this machine picks that out. It's amazing.
Anyway, I think we have a good beginning to have a good insight into what is causing this inflammatory disease and severe COVID. Very interesting.
Not clear why some people get the severe disease and others don't, because there's the potential for this to happen in anybody. Okay.
Could it be simply a matter of afucosylated IgG?
Who knows? It's probably other things going on.
Because remember, not all the infected patients have highly afucosylated IgG. Maybe there's a SNP associated with that, right? Oh, it's very interesting. Thank you, Amy, for pointing this out.
Oh, yeah. My pleasure.
It's a very nice paper.
It is a good paper. I'm interested in it, right?
Is there any evidence that enteroviruses do this?
Who knows? We have no idea.
You should look. You should look
Yeah,
one
I know.
day.
It's on my list. It's on my list of things to do in the
Dixon,
next 5 to 15 years of my life.
My camera's off.
What happened?
Nothing. I turned it off.
Oh, you don't want us to see you?
Okay. You can see me now.
Why do you just randomly take it? Are you taking a shower or something?
So my little grandson is here.
Oh, I'm sorry.
So,
I understand.
He knocks on the
Okay, I'm sorry. I just was wondering why you turn it off and on randomly.
Now
Why
you're
do you
muted,
need to know
Dixon.
everything?
I'm
Dixon.
I'm
He's
the
the producer
producer.
of the show.
I'm the
I don't want to
producer.
disturb the flow of action. That's why.
That's okay. I shouldn't have asked.
Don't micromanage Dixon. Let Dixon,
It's
the
okay,
star
it's
of
okay.
the show, have
No,
a
stop
little
it.
leeway.
Stop it.
Okay.
Yeah.
All right. Let us do some picks of the week.
Aha.
Dixon, what have you got this week?
Well, I have everybody's pick this week because Ketanji Brown Jackson got confirmed by a vote of 63 to 49, as I recall. But it was a little higher.
53 to 47.
53 to 47. I watched it be confirmed as the count was taken. It was a magnificent victory for diversity, for balance. It's amazing. And it's also amazing to see how many people didn't vote for her. And along party lines, they had no other reason than to say, well, the Republican Party told me not to vote for her, so I won't. That's about as non-thinking a person as I can imagine in government. So for two reasons, we celebrate her addition to the Supreme Court, which won't start until October. But I'm absolutely stunned by the result and encouraged. And she's highly qualified. I mean, highly qualified.
I just don't understand this herd mentality.
Well, she was so qualified that the herd mentality promoted her to her recent position like a year and a half ago.
That's true.
Which is amazing.
People who were for her during the interview systems now come out and say no.
That's what I'm saying, is that she got to
her idiocy. Sheer idiocy.
Well, as you know, and I don't want to get into politics, but the Supreme Court is supposed to stay out of politics, but it doesn't. And so if it can be skewed to one party, then favorable.
But this complaint isn't just about the Supreme Court. Like, so for instance, the person who was just approved the head of the FDA. He was head of the FDA by under Obama. He was approved by the Senate 99 to 0. Okay. And it's basically the same people who then barely approved him this time. And it was they needed Camilla, I think, to come and do the tiebreaker or it was pretty close to that. And his policies have not changed. He hasn't done anything, but it's just about the party.
That's exactly right.
Hasn't it always been, though?
Not so disgustingly.
Correct.
It's not about that.
And I'm looking forward to some interesting Supreme Court decisions that are split, not down party lines, but rather by philosophies, etc. Yeah.
So now, what is the liberal conservative count on the court?
It's the same. It doesn't swing.
It has not changed. Because the guy coming off is the same as the woman coming on.
Yeah, it's still, what is it, 6 to 3?
Yeah.
5 to 4?
Okay.
That's right.
I have to say, when I was younger, I remember when often votes in the Senate were like 99-1 because both parties could agree on things. And they can't seem to do that anymore because they hate each other.
Correct. That's right.
It's just unfortunate, and it's not the way.
But that's not true, though. They all voted for the stimulus package that that was passed before the Build Back Better one failed. So the infrastructure package passed by a very high vote, and it was almost consensus. So Biden has had a few victories among some of these horrible split down the middle party defeats. And the October elections, November elections of this year will show what really has happened. And I don't think we're going to be pleased.
We're never pleased.
I don't know. I'm pretty pleased right now. We have at least this ability to get somebody into the Supreme Court.
God, it was very close, Dixon. Very, very close.
It was.
By the way, speaking of, political party distinctions, I read an article about institutional biosafety committees. Okay. And I mean, I'm on the IBC at Columbia, and I happen to think it functions very well. But this article said basically they're obscure and nobody knows what's going on. Nobody knows about them. Well, if you just open your eyes and look, you'll find them. It's not hidden.
True.
So just, ignorance is not an excuse. Anyway, one of the people he spoke with was Richard Ebright, who is an opponent of apparently all kinds of good experiments because he thinks they're all unsafe. And he said he's had many inquiries from politicians about changing the system. And he said all the inquiries have been from Republicans. And so why do they want to limit experiments and Democrats do not? What's the bloody difference? Fundamentally, the outcome benefits everyone. So why do they need to regulate it, especially if they're not the party of regulation? I don't get that at all.
You want to go even further. Why do the Democrats always want to raise the NIH budgets, and why do the Republicans want to shoot it down? Because they're an anti-science group. That's why. They're not evidence-based. They're evangelism.
If I were evidence-based, it would be too hard for me to control people.
This is all true. Then you don't have control. You're just waiting for the results.
So, the problem is,
it's too willy-nilly. I don't have control, and I can't counter the argument.
And the problem
make
with
it
science,
a fearmonger thing.
Yeah, well, the problem with science is that it's neutral. It doesn't take sides on the results. So they don't like that.
Also the results.
Yeah, but science funding is not neutral. That's the problem.
No, it's not. No, it's not.
And the regulation of science is not neutral. So this is the problem. Right. But this is why I like to stay away from politics, because it's confounding. The science is beautiful, but the politics of science is horrible. And let other people
remember, though, without politics, you don't get your money.
Yes, we're well aware of that.
Do you know what happens if you mix politics with science, Dixon? You get politics. You get politics.
You get what? You get an unedible cake that no one wants.
All right. Thank you for your pick. Amy, what do you have for us?
So it's an article in The Atlantic that's written by the former Homeland Security director under Obama. And it's really a way to look at how we're going to adapt to the pandemic. And she talks about how, we fell in line with taking off our shoes at the airport for bioterrorism safety and stuff. And we were stringent about how much fluid you had to take on the plane and stuff. And now we've kind of relaxed all of those things to some degree.
And how we
adapt. And so it's just it's just a different perspective of how we're going to adapt to the pandemic And
I don't like that she calls the Omicron the hyper-infectious Omicron.
She's not a scientist, so she reads the news.
We did a paper showing that it's immune evasive. That's why
it doesn't matter what we did a paper on. She's a layperson. It's a tiny detail. The larger picture.
But you're missing
the message of what she's saying.
All right. So what are we going to do? We're just going to adapt to what? Wearing masks all the time?
Well, so that's what she talks about, is how we're going to learn to adapt to where we don't wear masks all the time and we become more normal and we don't have this population that's always in fear.
Look, we have
Just
influenza.
like we don't have a population that's always in fear of nuclear plants melting down.
Nuclear winter,
right? I mean, you had a population that did that. But was in fear after 3 Mile Island, and now we're done with it.
As we said earlier, we got used to 40,000 people a year dying from flu, right?
Correct.
We're going to get used to COVID, whatever form it is. Right. I totally agree. But right now we're not there yet. A lot of people are still scared.
Right. And so she just talks about it.
No, it's useful. It's useful.
But she's published it in a magazine that almost nobody reads.
Is that true?
Correct.
Why? Why not?
Because it's expensive and it's behind a paywall, and they only cater to intellectuals. This is a very, very high-end intellectual journal that has lots of heady articles, which is why Amy reads it, because she's a very intellectual person. And you get a lot of good views in it, but it's not a widely read.
It's $60 a year. That's not that much. It's $6 a month.
I don't know what their circulation is, but it's got to be low.
Oh, I don't know. They have a lot of authors. They have to pay them.
I don't know. Obama just had a recent article in it, and it was interesting too. And I think that might have been within there that I read the Bill Clinton article about his take on expanding NATO and Putin and where we stand today. I'm not sure if that was there or something or other.
I mean, they have a trio of reasonably good science writers.
Do I agree? I'm not criticizing that
because
at all.
of that, an enemies have picked some of their articles recently. I'm thinking of subscribing because I'd like to support them.
Sure. I look at it like The Economist. That's a very, very high-end journal also, and it's one that has a lot of great ideas in it. But it's read by a very narrow, focused group of people. And I'm not being critical. I'm just saying that if you want to get the word out,
I understand.
You don't publish it in the Atlantic.
I understand.
Yeah. According to this, in 2018, its total circulation was about 500,000.
That's not a lot.
No.
You guys ever hear of the publication called Foreign Affairs?
Sure.
It's just it used to be just a paper thing, no pictures, just paper, right?
Exactly.
And when I was in high school, I subscribed for a year.
Did you really?
I didn't understand. I didn't understand a word of any article.
Exactly right.
It was so hard to read. I probably would get it now, but I was
into Foreign Service.
I was into it. I was into that, and I thought I would really enjoy it. And man, oh boy,
I don't know, but I mean, I think like with writers of Catherine Wu and Ed Young and stuff, and I think their prominence on Twitter, I would not be surprised if it were an underestimation of the circulation. But what do I know?
Remember, I'm not being critical of the journal.
No,
I'm just saying if you want to get the word out, you don't publish it in the Atlantic.
Right.
Well, it's a magazine, right, Dixon?
Yeah, that's right. And when does it come out? Every month?
Yeah, it's monthly.
10 issues a year.
10 issues. They take a break.
You got the difference between Science magazine and Nature and the American Scientist or the Science Journal, the Science Diary. The moment you start popularizing an idea, the circulation has to go way up in order for it to make any sense.
Understood. Thank you. Rich, what do you have for us?
On a lighter note.
Sorry.
This is a local pick. It's the Lady Bird Johnson Wildflower Center. So the Johnsons, LBJ, and his wife, Lady Bird Johnson, were, sort of local heroes, having been raised, brought up nearby in Texas. And Lady Bird Johnson herself is an environmentalist type with a special interest in native plants and wildflowers. And I believe it was during her life that she established this wildflower center, which is basically a botanical garden. I think it was moved after she died and carried on by her one or both of her daughters. At any rate, it's about 10 minutes from where I live. And it comes to mind now for two reasons. First of all, this morning I went to the 2022 Molecular Biosciences retreat from UT Austin to hear a couple of talks that was done at the Wildflower Center. Okay, which is a great venue. And the other is that it is spring in Texas, which is a special time everywhere.
It is.
Special time in Texas as well. Dixon mentioned the bluebonnets earlier on. So I had a chance to not only go to these talks, but to visit the Wildflower Center in spring. And it's just absolutely gorgeous and a wonderful facility. They got a plant sale going on now. So if you're ever in Austin or if you live in Austin, I highly recommend it. And now is the time to do it because it's just gorgeous.
Nice.
I remember a quote from Lady Bird Johnson while she was still alive, as she was getting involved in the beautification of America. She wanted to plant all of these along the islands of highways because they'll be self-seeding after a while. The draft from the cars actually distributes the seeds. And her favorite quote was, "I'd highly encourage you to plant a tree, a shrub, or a bush." And she was vilified for that for a while, and all of a sudden she became everybody's hero. Even her husband was a little bit sick and tired of hearing about it, but she would come out on her porch. Rich, I actually had the privilege of waving to her once because I had a paramour that I was involved with who came from the hill country of Texas, Seguin, and she knew exactly where Lady Bird Johnson. So we went over to her house and we stood outside across the river and we waved. She came out on our porch and she waved back. "Are you planting a tree, a shrub, or a bush?"
You do a good accent, Dixon. Very good.
Well, she was easy to do, but she was a lovely woman with a very high level of sensitivity. And how she could put up with such a cantankerous old man like LBJ is beyond anybody's imagination. I'm serious about that. Their life was contentious.
Yeah, but she liked them, I guess, right?
They were in love, but it had a funny way of expressing itself.
He was something. But Dixon, you also do a good Bill Clinton accent also.
No, but please.
And you do a good Jimmy Carter accent too.
Well, again, I'm on hold for those performances.
I'm trying to get him to do it, but he will not do it.
No, I can't.
Back to old TWiV.
I cannot verify doing that accent on this show.
I cannot verify. All right. Thank you, Dixon. Thank you, Rich. And thank you, Dixon, for the color commentary. So I have a YouTube channel by a fella named Martin Doulard. And this guy, I think he's from the Netherlands. He's a graphic artist or something. He decided to buy a cabin up in the mountains in Italy and renovate them. And it's, he wants to create an off-the-grid homestead. There's no electricity. So he makes videos. And this is the thing that is amazing to me. He's doing all this work, right? They're stone cabins. The roof leaks. It's a mess because one of them was used for animals for years. And he can't even drive his car next to it because the road doesn't go up. And he wants to fix them. And one of the first things he does is put in solar panels so he can get electricity. And the winter is brutally cold. In fact, he's trying to work through the winter. And at night, he has to go stay in a local motel because it's too cold to stay in the cabin. And he puts a little wood-burning fireplace in. And he said, the problem is I sit next to this. There's no insulation in the cabin. There's no windows. He said, half of me is warm and the other half is freezing. But he just goes through, what he's doing. But what I find is cool is that he's obviously setting up video cameras on tripods. You never see them. And he's got to spend 20% of his time just getting the right shot besides doing the work he's trying to do, right? And I appreciate that. And then he says, at night, I go in my cabin and try and edit the videos if there's enough power left. The batteries tend to run out at night because there's no sun to charge them. So it's quite interesting. In one episode, he was building window frames because he wants to put windows in, and it's all stone. So, it's hard to put wood in next to stone. You have to, but he said, I'm sorry we missed part of this because the cameras stopped functioning in the cold weather, So I really like it for the video. He does a really nice job on the video, and he's way up in the mountains. It's spectacular. But I have to say, you'd never get me to do this. No. Never in a million years would I want to do this. It just looks painful. I don't know why he wants to do this.
He has 226,000 subscribers.
Yeah.
And the first video in this sequence has 3.3 million views. Unbelievable.
I'm not surprised. People kind of like this idea of going off the grid. But his videos are well done. They're very good, and they're very soothing. He's not contentious like me. He's not. He's just got a calm voice. When things go wrong, he says, "What am I going to do about this?" And he figures it out. He's got a lot of power tools. They're all battery driven because he can't put outlets anywhere. Anyway, I find it very cool. You might like it.
Here's another video.
I'm always worried about people like that because they're far away from medical, and if they get into an accident, they fall off a roof.
Yeah,
they drive a nail through their hand, or they slip and hit their head. They're in big trouble very fast.
Oh yes, because he'd be lying there for a long time. There's nobody around there,
and the video cameras would pick it up, but it would be too late.
Too late.
He's got a series of videos called 2 Years on a Bike.
Oh, really?
Apparently, he published a book about his adventure biking from Vancouver to Patagonia.
Cool.
Wow.
And there's, I guess, a series of 4 videos covering that. Amazing.
It's an interesting fellow.
Yep.
And apparently he does his work from home so he can keep working and get paid and pay for all this stuff. Anyway, it's kind of interesting. We have a couple of listener picks. One is from Anne, who writes, "This is a wonderful review by Sate and Crotty about the memory response to infection by SARS-CoV-2 and in particular about hybrid immunity. It's called Immunological Memory to SARS-CoV-2 Infection and COVID-19. I couldn't find the paper, the review. I emailed her. Hopefully by the time I put up the show notes, she'll answer because I could not find a review of that title. Let's see. Oh, here it is. She answered me. So I see why I couldn't find it because it is a preprint. It's Zenodo. Okay. Let's see if this works. Yeah, it's a preprint. Okay. Thank you, Anne, for sending that. How about that real-time listener pick? And we also have an email from Chris with regard to your recent TWiV with Brianne, in which you both lamented the lack of tools to characterize T-cell responses. This company seems to be working on innovative solutions to that problem. And he sends a link to a company called Isoplexis, who apparently is doing such things that related to T-cell measurements of T-cells. Let's see if they have a statement about us. I don't care about your management team. Our mission is to accelerate the fight against cancer. Okay. Okay, good. But I believe Chris when he says they're working on T-cell responses. Okay, that'll do it for TWiV 887. You can find the show notes at microbe.tv/TWiV. You can send us a question, comment, a pick to TWiV at microbe.tv. And if you enjoy our conversations, even if you don't, please help support us. If you don't like them, you're not going to support us. I know that. You can send us your contributions because we're a 501c3 and your contribution would be U.S. federal tax deductible. Go to microbe.tv/contribute for all the pertinent information. Dixon Despommiers at trichinella.org and thelivingriver.org. Thank you, Dixon.
You're welcome. Nice breadth and depth of subjects this time. Enjoyed them.
Very good. I will be lamenting the day you don't say that any longer.
That would never happen unless I was already recycling my molecules.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
And Amy Rosenfeld is at Columbia University, enterovirus.net. Thank you, Amy.
Thank you.
Where are you going next?
To the lab to do
Okay.
my
You're
ELISA?
supposed to. No, you're supposed to say Disney World.
Oh, Disney World. I'm going to Disney World.
Okay.
That's right.
Enjoy.
I'm not clear what's at Disney World besides Mickey.
Because of
and
the Super
Goofy.
Bowl. In the Super Bowl, when the quarterback who wins the game, then they come right up to him and they say, and now what are you going to do? And he says, I'm going to go to Disney World. They must get about $100,000 for saying that.
That's it?
Well, that's a lot for just those few words, don't you think?
Okay. Well, it's
So,
$100,000 that I got, then more than I just got paid.
You'll go to Disney World for that. Of course you will.
So
That's a whole grant.
it's like 3 months of a grant.
And they've got all the mice you could ever use, but they're all named Mickey.
It's okay.
Yeah, they're
It's
inbred.
okay.
That's good.
They're all inbred.
That's
It's
right.
all good.
They're-
I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.